Okay, we will have the final lecture of the morning. ¿Cómo está, Miguel? ¿Todo bien?
I'm very well. Thank you very much. How are you doing?
We are just doing fine. So I would like to thank you very much for accepting the invitation. It's a real honor for us to have you here. Well, Miguel is a professor of physiology in the University of Santiago de Compostela in Spain, and he's a brilliant investigator. He has been working with mechanisms that are sensors for nutrients in the hypothalamus and how these sensor mechanisms control thermogenesis in peripheral tissues. He has been awarded several times, most recently by the European Journal of Endocrinology, and he has more than 200 papers that are highly cited. So it's really an honor, and it's a very good collaborator. That's an important point here.
That's true.
That's the most important point here.
Yeah.
So I would like to thank you for accepting the invitation. You can share your screen, and once you finish the talk, we'll have some few questions for you.
Okay.
If it's okay for you, of course.
Yeah, for sure. Okay. It is okay now? Yeah. Can you see the screen?
Yes. Yeah, it's okay.
Thank you. I would like to start my talk thanking everybody in the organization of this meeting, and special thanks to Lucio, because we have been collaborating many years now, and I have the chance to have in my lab Natalia Dragano, who was trained by Lucio during her PhD stage, and Natalia is one of the best postdocs I have had in my lab over all these years. I became PI, and I really thank Lucio for that and for the inspiration of having her in my laboratory and this collaboration we have ongoing. Over the last 20-25 minutes, I would try to summarize what we have been doing over the last more or less 15-20 years, which has been mainly focused on AMPK and possibilities of target this molecule in the hypothalamus for obesity therapies. There is a pandemic problem in the world currently, a part of COVID, of course, and there's a clear increase in the obesity rate worldwide. No? Everybody here in this audience knows that, and therefore it is demanding to know the mechanisms modulating energy homeostasis. But there is a critical question here, and the next one is how this works. It's just a matter of thermodynamics, and basically energy balance or energy in the body results of a balance between the energy that we eat, the food intake, and the energy that we spend. If there is an imbalance between these two sides of this equation, then there is overweight and obesity. And the problem with obesity is that fat can be stored in several organs that are not prepared for dealing with this fat excess. For example, the liver, the muscle, or the pancreas, and many others. And this promotes metabolic disorders associated to obesity. Then it's very demanding to know how this energy balance equation is controlled. And for that, there are some key questions that need to be addressed. First of all, how is energy sensed by cells and the whole body? Second, how is energy sensed at the whole body level? Third, does energy sensing happen in the hypothalamus? And fourth, are these mechanisms a possible target against obesity, which is a relevant question because targeting things in the central nervous system has been a very complicated battle over the last 100 years or more of pharmacology. What is AMPK? AMPK is an energy sensor. It's an enzyme that is in almost every single eukaryotic cell, and is basically sensing the levels, an organism sensing the levels of energy in those cells. AMPK is complicated. It's a molecule which is composed of 3 different subunits, alpha, beta, and gamma. The subunit alpha is the catalytic one, and the beta and gamma are the regulatories. And there are several subunits, alpha 1, alpha 2, beta 1, beta 2, gamma 1, gamma 2, and gamma 3, all of them codified by different genes. The modulation of AMPK is a complicated process that depends of several upstreams, kinases and phosphatases, but there is a key factor modulating the activity of this enzyme and also controlling the activity of the upstream regulators. And this is the ratio AMP-ATP. The ratio AMP-ATP is a signal, is a indication of the energy status of the cell. If a cell has a high AMP-ATP ratio, this means that the cell is depleted of ATP and then it needs energy. And for that, AMPK gets active by phosphorylation of this threonine 172. And when this happens, AMPK at cellular level switches off ATP consuming processes while switching on ATP producing processes. For example, it switches off fatty acid synthesis and switches on fatty acid oxidation. This allows the cell to restore the energy levels. At the whole body level, AMPK have been demonstrating that it is doing basically the same. Is when AMPK is activated, he inhibits energy expenditure and increases food intake, increases appetite. We'll see now how this works in a few minutes, but I would like to tell you before that AMPK is not only an enzyme that is controlling fatty acid metabolism, which was the particular pathway in AMPK was first described. AMPK is basically controlling every single aspect of the cell metabolism. AMPK is controlling glucose metabolism, is controlling protein metabolism, is controlling, of course, lipid and cholesterol metabolism, and also mitochondrial biogenesis and autophagy. All these cellular processes are controlled by AMPK, because AMPK is modulating the activity of key enzymes regulating all these pathways. Then AMPK is a crossroads modulating, orchestrating the whole metabolic response of the cell by controlling this AMP-ATP ratio. This makes AMPK a very important molecule in terms of evolution. And in this sense, I would recommend to all people working in this field to read this paper, which is a paper, for Shivacher in this journal, Comparative Biochem Physiological Chem Biol, Mol Biol, that shows that the evolution of AMPK, and it can be seen in that paper, that it's a very nice review, that AMPK, the history of AMPK appears about 3 to 3.5 billion American billion years ago. This means that AMPK appeared in the evolution before that other important molecules and even important organelles that we have in the cell. For example, mitochondria, nucleosomes, histones, and much before that other typical hormones like insulin, the autonomic nervous system, any adipokine, thyroid hormones, or UCP1. This means that AMPK was already there when the cells were prokaryotic cells and the energy was not so abundant in the media they were living. There is also a very nice paper that I recommend everybody to read. This is this paper by Rostan et al. In the Journal of Experimental Botany. It's a journal about plants. And there is a nice phylogenetical analysis of AMPK, and it can be seen that AMPK, especially the AMPK gamma subunit, as well as some of its regulators, for example, the protein phosphatase 2 alpha, appear already, you can see here, in bacteria and archaea organisms, which is much, much before that, for example, mTOR or any other component of the cell metabolism, which means that AMPK was there long time ago, when someone is in one place in biological science, in biology, long time ago, and is very well preserved. This means that it is important. It's clear that AMPK is important for life because it's controlling metabolism and then every single aspect of the cell biology or the cell physiology, if we can talk about cell physiology, which is something that's always controversial because physiologists, as me, always like to talk about whole body level and everything. But anyway, AMPK is important at that level. And what is doing AMPK in the different organs or tissues that it is expressed? It's controlling several things. For example, it's important for the brown and the beige adipose tissue by modulating several processes there. It is important in the brain, controlling, for example, obesity and thermogenesis. You will see it controls also, for example, among other processes, fatty acid oxidation, mitochondrial biogenesis in muscle. It's important in the kidney, very relevant in fatty acid oxidation there. It's important in the heart. It's very relevant for the heart metabolism, in the immune system, in the endothelium of the blood vessels, and of course in the liver, which is the paradigm of metabolism at the whole level. Then MPK is clearly important. However, let's have a deep look to MPK in the brain, and in particular to MPK in the hypothalamus. The first paper that showed that MPK was important in the control of energy balance was this paper published in 2004 by Caroline Small and Dave Carlin, when they showed that AMPK in the hypothalamus was involved in the control of the food intake. Almost in parallel, a few weeks later, Barbara Kang in Harvard described similar findings, doing even some genetic modulation of AMPK at that level, and it was clear at that time that MPK regulated food intake also at hypothalamic level by controlling, responding nutrial and hormonal signal. The next key paper, in my humble opinion about that, was published by Dominic Withers and Mark Claret 3 years later, when they published in JCI that when they knocked down AGRP alpha-2 CYP unit in 2 different sets of neurons in the hypothalamus, AGRP or Ponzi neurons, the phenotype that they had were totally opposite. When MPK alpha-2 was knocked down in AGRP neurons, the animals were lean because they were eating less. However, when MPK was knocked down in the Ponzi neurons, the same arcuate nucleus, the animals got obese because they were eating more, which was clearly the first demonstration that physiologically the manipulation of the genetic manipulation of MPK was relevant for food intake control. We started working on MPK when I was doing my postdoctoral training in Tony Vidal-Puig laboratory in Cambridge, actually a long time ago. I started there almost 18 years ago. And we start to analyze the possible involvement of AMPK in the orexigenic action of ghrelin. And after several papers and the parallel work of other people in Santiago, Carlos Dieguez, Rubén Nogueiras, myself, Thomas and Horvath and Sabrina Diano in Yale, and Maria Casals in Barcelona also, Marta Corbonis at London. We showed that between all of us that basically ghrelin was acting in the hypothalamus to activate MPK. This involved a complicated downstream pathway that should modulate the activity of mitochondria leading to fatty acid oxidation, as well as the production of ceramides by CPT1 in the ER, and this in a double interaction cell. This was mainly sensed in our experience in the VMH, and this information was translated to the arcuate nucleus, where the expression of MPY and AGRP was increased and the food intake was increased. Then it was basically a mechanism by which ghrelin was activating feeding, or was promoting feeding by activating MPK in the hypothalamus. By that time, when we start working in that scenario, I was starting to set up my lab in Santiago in 2006. And at that time, I was mainly more interested in food intake, but I was working with Tony for these 4 years in Cambridge. And Tony is he has a very good lab in terms of thermogenesis and brain adipose tissue physiology. And because I was there 4 years, I was of course inspired by the excellent work he has been doing over all these years. And I started also to have a look to energy expenditure. And you know that energy expenditure has different components. And one of them is thermogenesis, which is quite important for homeothermic animals, because maintaining a constant the temperature of an animal of my size, which is about almost 90 kilos of body weight at 37 degrees or all my life in Santiago de Compostela, which it's raining all the time and it's cold, that spends a lot of energy. Maybe in Brazil is easier, or thermogenesis is not that important, but here in the north of Spain, this is cold, and we need to have good thermogenesis, not as much as Sweden, but thermogenesis is important for body weight control. Then the amount of energy that we spend maintaining our body temperature constant is important in terms of whole body energy balance. And the interesting stuff about brown fat, which put my attention back those years, and I have been keep working on that, was that the central relation of brown thermogenesis depends mainly on the hypothalamus. There are several hypothalamic nuclei and areas that are sensing temperature information, as well as several circulating factors, etc., that project to several nuclei in the brain stems, for example, the Raphael or the Oliva, and these nucleus project to sympathetic nerves in the intermediolateral column of the spinal cord that project to the nerves controlling the brown fat activity. For example, increasing lipolysis there, or increasing the expression of UCP1 by beta adrenergic 3 receptor signaling. Then controlling the activity of the hypothalamus can be a useful tool to modulate the activity of brown fat. That this can be achieved and has been demonstrated many times. Then we start working in molecules that we knew that they have very potent thermogenic action, but most of these molecules were aside before we start working on that peripherally. Everybody knew that when T3 or T4 thyroid hormones were given peripherally, you had thermogenic action. But we wanted to know whether that effect could be dependent of central actions of the thyroid hormone. This means not the thyroid hormone acting directly on the brown fat. The thyroid hormones go to the hypothalamus and signal there through the sympathetic nervous system to activate brown fat thermogenesis. And basically, when T3 was given centrally, ICB or in several hypothalamic nuclei, we observed this kind of phenotype. The animals lost a lot of weight, and that weight was feeding independent. And that happened with T3 was given in the VMH, but not in other hypothalamic nuclei. For example, in the arcuate nucleus, it was pretty specific, a weight-dependent induced, a food intake independent induced weight loss. No? In collaboration with my long-term collaborator and colleague, Kamal Ramouni in Iowa, we also demonstrated, these are data from his lab, that the activity of the nerves that were innervating the brown fat was very dependent of the administration of T3. That was a very clear dose response data showing that the sympathetic firing of these nerves is increased. The activity of the nerve is increased after the T3 administration. Moreover, in association with all this data, we had that when T3 was given centrally, this was ICV or in the VMH, we had a marked activation in the expression of thermogenic markers in the brown fat in terms of mRNA and also protein. In this case, UCP1 was increased in the T3 treated animals. And finally, we had some functional data in collaboration with our imaging unit here in Santiago, and also people from Bilbao, showing the Patricia Spichueta and Malu Martinez Chantal, showing that the activity of the brown fat in these PT-CT scans was clearly increased when T3 was given centrally. You can see here how the glucose uptake in the brown fat area increases after T3 administration, as well as the oxygen consumption by the brat itself or the isolated mitochondria from the bat of those animals. This led us to postulate about 10 years ago a model which was improved in 2017, showing that T3 was acting in the hypothalamus to modulate the activity of AMPK alpha 1, and this promoted changes in the levels of ceramides, leading to ER stress decrease, which was the responsible of the sympathetic innervation of the brown fat, leading to increased thermogenesis, and also the activation of YANG1 at that level promote the parasympathetic drive to the liver that was creating the fuel, the lipids necessary for the brown fat to be burned and obtain that energy. This was a mechanism that we proposed that time. And the first question we had was, is this something specific for T3? And is there any other signal involved in this metabolism? And we have been investigating this for the last 10 years. And we have seen that this is a canonical pathway. And basically, the inhibition of AMPK in the VMH, whatever the stimulus is, it can be estradiol, it can be BMPAV, it can be nicotine, it can be liraglutide, and also leptin, which has been published for another group. All these factors promote inhibition of AMPK in the VMH, increase in the sympathetic tone to the brown fat, increasing the expression of thermogenic markers at that level, for example, UCP1, increase of thermogenesis and feeding independent weight loss, which means that we are able to modulate weight loss independently of feeding by controlling the activity of AMPK in the VMH, in that particular hypothalamic nucleus. This is a canonical pathway. We have proposed this several years ago, and basically any single factor modulating AMPK at this level promotes back thermogenesis and browning. It's important to know that, remember this, Mark Claret and Dominic Whittier demonstrated back in 2007 that AMPK inhibition in the arcuate nucleus promoted or modulated, AMPK in the arcuate nucleus modulated food intake, and the inhibition of AMPK at that level inhibits food intake. Then we have the same molecule in two different nuclei, the arcuate and the VMH, controlling the two key aspects of the energy balance equation, feeding and thermogenesis, and then energy expenditure. We wanted to go further in the knowledge of AMPK, and we have been generating AMPK models in the hypothalamus in the last years, in the last 5-6 years. The first of all is the BMH specific knockout that we developed using the LOS-P-CRE technology by crossing LOS-P AMPK alpha-1 mice with mice expressing SF1 and CRE under the control of SF1. SF1 is a particular cell population within the VMH. Actually, not all the cells in the VMH are SF1 positive, but a high percentage of VMH cells are SF1 positive. Not all of them, but a considerable amount at that level. And basically, when AMPK alpha-1 was ablated in these SF1 neurons, we had a lymphophenotype characterized by lower adiposity, no changes in food intake, and no really significant change. Sorry, no changes in body length and no changes in food intake. The phenotype of this animal, the lymphophenotype, relay in a higher energy expenditure, as you can see here, than the expenditure of the knockouts. It's much higher than the wild-type littermates, and this was totally associated to a marked increase in the thermogenesis of these animals in the brown fat. The expression of thermogenic markers is highly increased in the brown fat of the knockout, as well as the temperature of the brown fat area. Then we have a model in which we recapitulate the effect of thyroid hormones, estradiol, BMPB, nicotine, liraglutide by modulating the activity of AMPK, specifically in this SF1 cell population within the VMH. You can see here also the PET-CT scan, and you can see here how the glucose uptake, the labeled glucose uptake in the brown fat of the knockouts is much higher than in the flux wild-type animals. The quantification, but it really doesn't need too much quantification. It's very clear if you look to the images. Then, this is interesting. And so far in the lab, we have 5 more hypothalamic models of AMPK in several nuclei and several isoforms. Actually, 4 of them, we have even more, I think, are not published so far. And 4 of them, they have a very clear phenotype, as well as the one that Dominic and Witters and Mark Claret published in 2007. And this is interesting. I enjoy, but it's nice. The data I'm passionate of this, and I love to work in this field. But in terms of practical point of view, the key question here is whether AMPK in the hypothalamus may be a realistic target for the treatment of obesity. It's just incremental knowledge, which is good and I love, but it may have any possible application to improve the health of people, which is a very relevant question worldwide currently. Then the first target that we have looked for is AMPK in the VMH. In theory, if you inhibit AMPK in the VMH, you will have an activation of the sympathetic tone to brown fat, leading to increased thermogenesis, and also browning, something is beeping here, browning of the white fat, increasing energy expenditure and leading to weight loss. Then, evidence this one. If you have overactomized rats, which are obese, and you treat them with adenovirus encoding dominant negative isoforms of AMPK within the VMH, you have weight loss, which is independent of food intake. And it happened the same in diet animals. If you have obese mice and you treat them with this AMPK alpha-1 dominant negative isoform, body weight loss is achieved without major changes in feeding. If we have a look to the phenotype of the AMPK alpha-1 SF1 cre mice under high-fat diet, they are lean. They are leaner, sorry. You can see the body weight. And they are eating the same. And importantly, and again, this is based in a higher thermogenic response. These animals have higher temperature of the brown fat area and higher expression of the thermogenic markers in terms of mRNA and in terms of protein in that brown fat. This means that we have a model where using genetic manipulation of AMPK in the VMH, in this case, in SF1 neurons, we have reversed the obese phenotype in these animals. And importantly, this is also associated with that better glucose homeostasis in that mice. These animals deal much better with glucose than the obese wild types, which means that modulating AMPK in the hypothalamus can be a possible target to treat obesity, because targeting AMPK in the SF1 neurons, we can control thermogenesis, and targeting AMPK in another nuclei, for example, in Ponzi neurons in the ARC, we can control body weight, food intake, sorry. Then the key question here is, would it be possible to specifically target AMPK alpha-1 in SF1 neurons, and more importantly, using a peripheral treatment? This is an injection, a pill, or something. Of course, we cannot treat obesity in people doing a stereotactical injection and introducing adenovirus at that level. Then we propose this strategy in which we are working now extensively, and is the look or the research in neuronal targeted carriers that, using peripheral administration, can allow us to reach the brain, to cross the blood-brain barrier, and modulate specifically the activity of AMPK in the VMH in this case, or in the arcuate if we target the Ponzi neurons. And we will do that, or we are trying to do that, using specific promoters that we know that are expressed in those specific set of neurons, for example, SF1 or Ponzi, in the case that we want to target a food intake. Then, just to finish, the take-home message of this talk is that a hypothalamic AMPK is a canonical modulator of energy balance by controlling food intake, thermogenesis, glucose and peripheral lipid metabolism, as well as carbohydrate preference. A recent paper published in 2017 showed that nicely, published in some reports. AMPK increased the carbohydrate preference in comparison with lipids. And I'm positive that targeting of AMPK in the hypothalamus can be a realistic target for obesity in the future. We will try to address this in the next years and the next work to come in my lab in Santiago. I would like to thank again the organization. I would like to thank all the collaborators I have had the last years, people in working the AMPK topic specifically, the funders, of course, people in my lab. Special thanks to Edward Milbank and Natalia Dragano, which are the postdocs in the lab, and how they are leading this research on AMPK, and former postdocs like Pablo Blanco, Patricia Suani, Nora Martinez, Luis Martins, or Cristina Contreras, as well as PhD students. My colleagues in Santiago, Rubén Nogueiras and Carlos Deguez, colleagues and friends. I've been working for them for the last almost 25 years. Manuel Tena-Semper in Cordoba, which all the interaction between AMPK and the gonadal axis, as well as the interaction between the gonadal axis and ceramides. Kamal Ramouni in Iowa, who is the master of the sympathetic nervous system. Frances Villarroya in Barcelona, and Barbara Cannon and Andadegar in Sweden. They are the masters of the brown adipose tissue. Carmen Martinez and Naina Andarhisto-Haina in Angers, working about possible ways to target AMPK in the hypothalamus. In Munich, Matthias Schopp and Cristina Garcia Cáceres, which we are collaborating then in the role of AMPK in another central nervous system cells. In Cambridge, Tony Vidal-Puig and Chris Chattery, and in London, Dave Carling, which we are alongside Graham Hardy back in the 80s. He basically cloned and discovered, among others, AMPK. This is our research building where we work, the CIMUS in Santiago. My lab, my office are around that level when we spend most of our time. In the city of Santiago de Compostela, we have a very old university, it's more than 500 years old, a big cathedral, a lot of churches. You know that Santiago de Compostela is a place for pilgrims, for Catholic pilgrims, and excellent seafoods and excellent weather. It only rains once a year. It starts in October and finishes in May. It's a fantastic place for if you want to see rain. This is a perfect place to visit. I hope to see you here at some point with this situation. Hopefully we'll finish soon. Thank you very much again. This has been a real pleasure. Thank you, Licio. Thank you very much. I really appreciate it. Thank you.
Thank you, Miguel. You were just about to convince me that I should go immediately to Santiago, except for the last part of your explanations.
You get used. You get used. I think that actually Natalia prefers Santiago's weather than Campina's weather, I think now, after so many.
Okay, thank you very much. This was a fantastic talk, very enlightening. And we have some questions. Are you up to answer the questions?
Yeah, sure.
Okay, so the first one comes from Professor Marcio Torsoni. Marcio Torsoni is a professor of biochemistry at the University of Campinas, and he asks if AMPK alpha 1 and alpha 2 have the same expression in different regions of the hypothalamus, and if the effect on thermogenesis was detected on alpha 2 as well. So the differences in expression in different regions of the hypothalamus, and if you have looked into the impact of alpha 2 in thermogenesis.
These are very good questions. AMPK alpha 1 and AMPK alpha 2 are expressed both in all the hypothalamic nucleus we have aside them, we can detect them, but the interaction with the sympathetic nervous system and the control of brown fat thermogenesis is different. Alpha 1 is the isoforms, which is more involved in the control of thermogenesis. Alpha 2 doesn't regulate too much thermogenesis, and in our hands and in the model that we have generated, alpha 2 is likely most involved in the modulation of food intake than thermogenesis. Yeah, that is a very good question. Thank you.
Thank you. So now we have a question from Felipe Mosovic. He's a student at Marcelo Mori's lab. Marcelo Mori is working here at the Institute of Biology. Probably you know him. And Felipe wants to ask, I'd like to ask whether targeting hypothalamic VMH AMPK would affect reproduction. Have you evaluated any reproductive aspect of this model?
That's also a great question. Thank you for asking me that. We have been working with Manuel Tenasemper in Cordoba, and we have seen that knockdown of AMPK in different important neurons in the HPG axis, it has an impact on reproduction. For example, in GNRH or KISS neurons, in both sets of neurons, AMPK is important. In the VMH, we have not done a deep study of the reproductive function in these animals, but they look normal. I guess this question is based on the fact that alteration in the VMH function can have a behavioral can affect behavioral aspects of reproduction. We haven't checked that, but in terms of number of litters, size of the litters, fertility of the animals, they look normal when we treat AMPK alpha 1 SF1 neurons. We haven't seen any different, but it would be interesting to check, of course, of this mating behavior and this reproductive behavior that the animals show when other VMH population are targeted. Yeah.
Okay. Next question comes from Alexandre Moura. Alexandre Moura is PhD student at my lab, and he was recently on a training in Jeff Friedman's lab in New York.
Wow.
So he asks if the thermogenic response to hypothalamic AMP inhibition is also observed in the inguinal what?
First of all, congratulations for being in Jeff Friedman lab. It is one of my heroes. I have two heroes in my. I have too many heroes, but two of the people I admire the most are Jeff Friedman and Jens Bruning and Ronald Kahn. I really admire all those guys because they keep going all the time, and what they do is absolutely fantastic. I had the chance to meet Jeff some years ago, and was one of the experiences of my life. We have detected changes after the manipulation of AMPK in the subcutaneous and in the inguinal brown fat, but mainly most in the subcutaneous. But the effect of thyroid hormones on browning, mediated by AMPK, affect to both white fat deposits. Yeah.
Okay, we still have two questions. Is it okay?
No
for
problem.
You?
Happy, always
right.
happy to. Yeah.
So that's from Narayana Fanzolin. She asks if, in your opinion, what is the optimal strategy to maximize benefits to develop acutely therapy on hypothalamic AMPK? So I would like to put one part of my question within this, because I think this is very important. Do you believe the strategies that Richard DeMarchi used to construct molecules that can do 2 or 3 functions at the same time would apply in this kind of situation?
Yeah, I think so, because if the possibility of develop an AMPK modulator that could control the activity of AMPK in several hypothalamic cells can be of a real interest for that. Yeah. Actually, I'm pretty sure that some of the compounds that Richard DeMarchi and another of my heroes, Matthias Schopp, have been developing over the last years are working on hypothalamic AMPK because they have been targeting thyroid conjugates, estradiol-based conjugates, GLP-1 conjugates, and all these molecules are known to be acting on AMPK in the hypothalamus, and it is likely that they are targeting those cells and these mechanisms using the conjugates. In terms of the question that the other person, sorry, I didn't get the name, I apologize, asked, was that I'm not really sure if we are need to design a acute or a chronic strategy. I don't think that the obesity treatment can be based in a solid acute strategy because of the fact that can be compensatory or rebound effect. And if you target one side of the energy balance equation, automatically they only would compensate. Then I think we need to think in a chronic strategy that could allow us to modulate specifically hypothalamic AMPK without touching the periphery. We need to be quite refined there and try to look for mechanisms that can allow us to do that, control hypothalamic cells without peripheral effects.
Perfect. So last question, and then we let you go to your work.
Happy.
So that's from Marcela Raymond. She works as a master degree student at our lab here. In regards to thermogenic photos, do you follow an acute cold exposure protocol to CBAT activation?
No, in the images that I shown, all the animals that the images that I've shown in this talk, they were not animals exposed to cold. We have done experiments of thermoneutrality and cold exposure here, and we have looked for there's another paper that's almost ready to go that we examine the role of the physiological role of AMPK in several cold settings using several AMPK models. Hopefully will be ready to be submitted by, I would say, a couple of months, 3 months. And in that particular setting, we have used every single cold exposure we could imagine. And yeah, in that paper coming, we did it. But in the pictures that I showed, no, they were not cold induction there.
Well, I have one question regarding that. What is the room temperature that you adopt for your mice?
The normal room temperature for housing, I would say 22, 23, 24 degrees. When we do cold exposure, it depends if acute or chronic, 4 degrees. If you go for thermoneutrality, 29, 30. more or less, yeah.
Okay. So thank you very much. This was fantastic.
Thank you to you.
I hope next time you could come here. And
hopefully,
I hope I can go to visit you. I know the food is excellent and the churches are beautiful, and the rain, well, we can do with the rain.
But it rains once for October to May. If you can between June and September, should be fine. And if I go, of course, I hope you also will come to visit us, because I'm looking forward to meet you in person. Thank you very much for this opportunity to show our work, and special thanks also for giving me the chance to work with Natalia, and having her in my lab. It has been one of the best decisions of my professional career, and I owe you one. Thank you very much for everything, especially for that. Thank you.
Okay, Natalia was specially invited to join the room, so just say hello, Natalia. We want to see you.
Hola, Natalia.
Hola, Natalia. ¿Cómo estás?
Hello.
Hola. ¿Me escuchan?
Sí, sí.
Yeah.
Hi. Here.
Hi.
Better.
Okay.
I mean, working and listening.
I hope I can visit you both soon.
Hopefully.
Yes, you have to come, Licio.
Bye-bye. Thank you, Miguel.
Thank you. Thanks to you. Always. Thank you very much. Thank you. Bye. Bye. Bye.
Bye. Ok, para o pessoal que está ligado ainda, a gente faz um intervalo agora, e exatamente às 2 horas nós estamos aqui para a palestra da professora Carol de Michigan. Então vamos lá, almoçar rápido e voltar.